In a world where data science holds the key to addressing humanity's most challenging issues, the Data Science Without Borders DSWB initiative has emerged as a bridge across Africa and beyond, linking countries to opportunities that would help governments harness the power of data for positive change.
Data Science Without Borders, starting from its name, is a very interesting project. I'm the lead with a couple of colleagues from very many partnering institutions. It's a dream that we wanted to see data science spread across Africa. We always know that Africa has been known to lag behind the Industrial Revolution, but with the advent of AI and data science, we wanted a project that will bring partners in the African continent. We'll look at institutions coming together to start to harness the power of this new technology, to leverage their data sets, and to build capacity in data science and AI to help Africa realize its dreams through data.
Launched in February 2024, DSWB continues to empower African scientists to champion data-driven solutions. By breaking barriers and strengthening systems, DSWB is driving innovation and creating lasting impact across Africa.
Many times it's been said Africa needs to leapfrog. Yes, we need to leapfrog. We need to catch up with AI. We shouldn't be left behind, but we need to fix the processes that will get us there. Look for the data. Where is it? Is it in good quality? Do we have the people who are going to mine this data, analyze the data? Do they have the skill sets? Do we have the infrastructure to actually do the analysis and punch all these analyses out there? And of course, the governance. You know that Africa has a lot of data governance, diversity. How do we get to that point? How do we learn from the different countries? And once we fix these important pillars, even as we leapfrog, it will be the successful, effective use of data science and AI in Africa.
With passion, commitment, and collaboration, we can continue building a future where borders do not limit progress and where data science empowers us all.
So just one last housekeeping thing that I forgot to mention. If you need language interpretation from English to French, we do have language interpretation and interpreters in the call. So make sure to use that feature. And if you're also joining after this, after we have already started, we have an icebreaker question on line 29 of the Frama part. Please do tell us where you're joining us from and what your name is, and answer the questions that are on line 30. So we're going to go directly into the speakers sessions for this webinar. We do have two speakers for this particular session, Dr. Gabriel Rink and Dr. Sumir Panji from Eliwazi. We will have our first talk by Dr. Gabriel Rink, who's on the call. To introduce her, Dr. Gabriel Rink worked on a molecular epidemiology study on drug-resistant HIV at Public Health England early in her career after a PhD in virology. After a decade in the molecular diagnostics industry, leading the development and diagnostics and coordinating testing services for clinical trials, she joined the Wellcome Institute, where she managed a UK-wide genomic study focused on prenatal diagnoses and rare diseases. Since 2020, she has been at EMBL EBI, initially as part of the European Nucleotide Archive team. Her role revolved around COVID-19 related cohort data and integrating multi-omics datasets. Last year, she transitioned to the European Genome Phenome Archive team as the project lead for Federated EGA. Dr. Gabriel, it's my pleasure to welcome you to give your talk on fair data sharing and federated analysis.
Thank you very much for inviting me and giving this introduction. It's great. And I hope you can see my screen now. Just move.
Yes, I can see your slides, but they're not on present.
Yep. Should be 1 second. Okay, let's see. Okay, now it's probably better.
Yes, good.
Great. Okay, good. Yes, so my talk will span, on the one hand, what I've done in the past in the ENA team at EBI, but also what I'm currently doing with Federated EGA. And so it's all about pathogen data, cohort data, and this federated network for sharing human data. And okay. Yes, so this is about the resources, the portals we've built, how we created a process for linking data across different data types. And I thought the Contagio project might be of interest to you. And obviously, I will be talking about the federated network of EGA. So I don't know if you have come across EBI before. We belong to the European Molecular Biology Laboratory, which has 6 different sites within Europe, and we are one of them, based just outside of Cambridge in Easton in the UK. And we are the site that is focused on bioinformatics, and we host a whole range of archives for archiving various different types of data or biomolecular data. And at the core of our services that we provide is open science and the fair sharing principle to make data as open as possible, but also as closed as necessary. And yeah, it's a widely used service that you hopefully have come across in the past. So as I said, we have quite a large number of different archives that we host, and this covers everything from chemical molecules from drug discovery to nucleotide sequencing, proteins, structures, and imaging, disease-specific resources, but also resources linked to literature and general knowledge management. And so these are all archives hosted by us. And during the pandemic, there was a need to quickly access data for SARS-CoV-2, anything relating to COVID. And therefore, we have built a portal that sits on top of all of these repositories that I've just showed. And it actually can mobilize the data from all these different repositories into like one portal that can present all the different data types, everything that was relating to SARS-CoV-2 during the COVID-19 pandemic. But then we came to the end of the pandemic and we started thinking about, okay, we have created this platform, but now it's more about preparing for the next pandemic. And therefore we created the pathogens platform. And this is being led by my colleague Nadine Bachmann in the ENA team. And so the platform itself consists of, on the one hand, the pathogens portal that presents all the data related to pathogens, but it also has an aspect of providing pathogen data hubs that can be used by groups, for example, consortia, where data can be shared just within the consortium. The data will be kept private as long as it's necessary, so that it can be shared between researchers, and it also provides tools for analyzing the data. And there are now also national nodes for the pathogens portal in several countries that are all connected to our central pathogens portal. And later I will also talk about the cohort browser. Here is a bit more information about these data hubs. As I said, they provide tools for submitting, analyzing, visualizing, searching, and retrieving data. And within those data hubs, the data are kept privately and can be analyzed in the context of the whole ENA data repository. So with related data that may already be public. And currently we mainly have data hubs for foodborne outbreaks, for infectious diseases and antimicrobial resistance. And as I said, this initially was created in the context of SARS-CoV-2, but now it was sort of expanded to other infectious disease outbreaks with a view to pandemic preparedness. And as I said, it's for researchers who want to collaborate, for consortia, and also it may be of benefit perhaps for those researchers who may not have access to any compute environment where they can carry out the analysis. And there is a link later after the talk. We can share my slides, and because I've tried to include as many links as possible in my slides, and then you can have a look at anything that interests you in a bit more detail once you get access to the slides. Then I mentioned the pathogens portal, which retrieves information from the underlying repositories within the EBI infrastructure. And here we have sections covering different aspects, for example, nucleotide sequences. So someone has his microphone on. Proteins or wastewater, and also the cohorts browser that I've already mentioned. And so this now covers over 200,000 pathogen species, both from human pathogens, but also animal pathogens. And these are pathogens that are listed on the Health and Safety Authority list within the UK. This is how we sort of selected the pathogens that are included. And there is also a section specifically for those pathogens who are highlighted by the WHO as priority pathogens. And the national pathogen portal nodes that I've mentioned earlier, these have been created in Spain, Norway, Sweden, Switzerland, the Netherlands, and also Costa Rica. And but I've already mentioned, I also wanted to talk about cohort data. So why were we particularly interested in cohort data? The cohort data sets usually cover several data types, which could, for example, be pathogen sequences, but also information about the host, possibly clinical epidemiological information, or host genetics, or any other lab values. And it's really important to bring all these different data types together, because they represent a very valuable resource. And in many cases, these different data types are siloed in different repositories, and there is sometimes a lack of interoperability. So our goal was to bring all these data types together. And another aspects to remember in this context, obviously, is human clinical data. And there we have to think about data protection and compliance to all these data protection rules, so that we can keep the data safe. So we have been working for a few years. We have been working on a project called Recoded, which came to an end about 1.5 years ago. And here we established a data submission process and a way of linking the different data types together. And this is using the resources that are hosted within EBI. So you start off with your cohort data set. And then this, depending on the data type, then feeds into the dedicated repository for this particular data type. For example, viral sequences would be deposited into the European Nucleotide Archive, which is an open archive for sequence data. for example, for pathogens or animals and so on. And then, for example, if you had serology data, this could be reposited into biostudies. And if you wanted to capture a virtual biorepository, this would go into an archive called Biosample Archive. And then for the metadata that describes the cohort, this could be captured in the pathogens portal cohort browser. And then, as I said, for the human sensitive data, we have to consider data protection aspects. And for that reason, we have one repository which is specifically dedicated for hosting sensitive human data. And this is the European Genome Archive, or short EGA. And this operates a controlled access mechanism, which I will explain later in more detail. And then, as part of this project, we came up with a process how we can link these different data types on a participant level. And then, yeah, I've already mentioned the cohort browser. Again, this is a slide that you can look at after the talk, where these are all links to the different resources within EDI. You can just click on the links and look at the resources in a bit more detail. Coming back to the European Genome Archive that I've already explained, the archive dedicated for human sensitive data, and with the controlled access, where the data provider has a data processing agreement with EDI and CRG, who jointly operate the EGA. Then if a user would like to request data, would actually contact the data access committee, which is actually being set up by the data provider. And that way, the data provider remains in total control over who gets access to that data. So the data provider sets up the data access committee, who will receive the data access request from the user. And then a decision is taken by the data access committee if it should be approved or not. Once the request is approved, a data access agreement will be put in place between the data provider and the person requesting access. And once this agreement is signed, then the archive would release the data to the user wishing to access the data. And when we set up this process as part of the Recoded project, we had pilot study data set that we put through the process. Here in this data set, we had viral sequences that were deposited into ENA. We had T-cell and B-cell data and also antibody profiles that were deposited into the relevant archives. Biostudies and ARIExpress and sensitive data, as I've explained, went into the controlled access EGA. And then if you are interested in a bit more detail, there was a paper about the experience of the data provider, all the hurdles and challenges that they were struggling with throughout making the data available, like collecting the different data types from different departments within the hospital and so on. And also we described the process in a showcase on the Infectious Disease Toolkit website, where again you can read up a bit more about the process that we have implemented for this. And this is just a different representation of this process. As I said, the clinical epidemiological data went into the EGA archives, and the viral data into ENA, and the antibody profiles, B-cell, T-cell data into biostudies and ARIExpress. And then about the linking. For the linking, we're using the archive biosamples. This is an archive that documents the sample itself, and it can also capture relationship between samples. So the way we have created the link between the samples is we have created a record in biosample that represents the patient from which the samples were originating, and then each record within the other archives again created a record within the biosamples database. This could be sort of different data types, or it could be sort of the same data type but different time points of the same data type. And then there's an attribute called derived from, which then captures a relationship between those. And that way we created the link on a participant level between different data types. And then bringing it all together and representing this visually, we have the cohort browser, which then describes the cohort itself with the relevant study level metadata, where available some aggregate data about the clinical epidemiological information. It has a search and filtering functionality. It's fully integrated into the pathogens portal, and it also provides the data sets. And this is how it looks. So here you can see the metadata information about the cohorts. Then and then you have search and filtering features in the cohort browser. You can choose which columns you want to view. And also there is here a column that shows which type of data were collected for a particular cohort. And in the last 12 months, we have increased the number of cohorts listed in this cohort browser from initially just the one pilot study to 19 cohorts now. You can also list your cohort in this cohort browser if you haven't submitted the raw data and all these different data types to EBI. And in these cases, we just symbolize this with these donut icons. And in those cases where we actually have the raw data or the different data types saved within the EBI archives, we actually have hyperlinks that when you click on those symbols, it takes you to the relevant data set within the archive. And as I said, we use the biosamples archive for creating the link. And so here, this is how it shows in the biosamples archive. You can sort of search with text for the data set you're interested in. It sort of breaks it down into sort of filtering functionality here on the side. And this, for example, is patient number 1 of this study. And then it lists all the data points that we have available for this particular patient. And also it has links to the relevant data sets, which include samples of this particular patient. Then we have also added a dashboard to the cohort browser, which shows each cohort in a bit more detail. This is again the metadata information and also some statistics and the location. So this is the data, the view of the cohort level metadata. And then this is for the statistics, where it sort of breaks it down by type of sample and the gender. And here this table again lists all the connections. And here the Venn diagram and also this diagram shows which data, how many samples have how many data types connected, because not every patient has every single data type collected. And then there is also a feature with a map where you can see where these samples were collected. And when, as I mentioned, all this work was initially established with the Recoded project. When the Recoded project came to an end, we started a project called Contagio, and which continues from the Recoded project in that we can sort of continue with this submission pipeline workflow. And the idea of this Contagio project is to create what they call a cohort warm base, because during the COVID-19 pandemic, it became clear that it actually takes quite some time to set up a cohort from scratch. And if you are in a situation of a pandemic where decisions need to be made quickly, you don't actually have necessarily the time to wait for the whole setup of a cohort. And therefore, the idea is to create sort of a small-scale cohort that runs in the background. And then if a future outbreak occurs, these cohorts can be reactivated more quickly than starting a cohort completely from scratch. So the European Commission kindly made available funds to have two of these projects. One is Contagio, and another similar project is called Connect. Connect is focused on purely Europe, whereas in Contagio we have many partners from the global south. And so Contagio is mainly focused on arboviruses, for example, Zika virus, the dengue virus, and so on. And the idea is to create a roadmap and a toolbox to help reactivating these cohorts and providing tools for interoperability, creating electronic case report forms, and tools for data sharing, like this data sharing process that I've talked about. And also it's setting up a cohort registry in work package 1, which doesn't capture the information about certain data sets, but it captures information about cohort sites and their capabilities and expertise. And it also looks at ethical legal challenges in this context, and also it looks at virtual biorepository. And so this is this cohort registry, which is work being led by Adriana Tami, based at Groningen in the Netherlands, and also she is also based in Venezuela as well. And here, this is capturing sort of more the capacity of the cohorts where they are based and their expertise and PIs that are key to these cohorts. And these are institutes, as I said, Contagio is mainly focused on the global south. And these are institutes that they are working with for this cohort registry. And the other aspect was the virtual biorepository. And in the virtual biorepository, again, during the pandemic, it became clear that it's sometimes difficult to be able to access relevant samples, and you're sometimes not quite sure about the quality, or there may have been issues in the past with sending samples to other countries and so on. And in this virtual repository aspect of the project, this is looking at the practicalities and implementing appropriate processes and sort of going through the processes, identifying hurdles and how they can be overcome. Like, for example, what needs to be done to ship samples and so on. But this is on a very small scale with only 10 different sites, and each site is only collecting like 10 samples. So it's really a very, very small scale project. And so the planning has already started during the Recoded project, but then when Contagious started, they have selected these 10 sites to join the virtual biorepository, and where these 10 sites then work together towards establishing the necessary process. And then in the future, they are planning to widen this and to include more and more sites to this system. And currently, these are the 10 selected sites, and these were selected based on their expertise, but also to ensure a spread across the globe. And again, with focus on the global south as well. But now, let's come to the Federated network, the Federated European Genome Archive. This is what I'm currently working on. And so earlier I talked about the Central European Genome Archive, EGA. And this was initially started by EVI in 2008. And then in 2013, CRG, a site or an organization in Barcelona, joined forces with EVI to jointly manage the European Genome Archive together. And this is our activities so far. So in total, the EGA hosts data that add up to nearly 20 petabytes in storage and over 4 million files in total since the beginning in 2008. But if you look at the data for just 2024, we released data of over 1.3 petabytes, and we have thousands of requesters and data sets that are being released every year. And but then about 12, 15 years ago, or 12, 10 years ago, the idea of sort of a federated approach to dealing with human data emerged. And then in 2016, the concept of a federated system for this European Genome Archive was born. But then it took a few years, then until another 5 years for the first demo of this new concept. And then in 2022, the Federated EGA was officially launched, where 5 national nodes actually signed all the legal paperwork and were necessary around this setting up this network. And these initial nodes were Finland, Germany, Norway, Spain, and Sweden. But since 2022, a few more nodes have joined the Federation, and we now have 9 national nodes. And at the end of last year, we had the Canadian node join our Federation. So we have spread beyond Europe now. And we are hoping to further extend our network and have more nodes all across the globe. And since the beginning of last year, we also have the first data sets actually available within Federated EGA. So this is Federated EGA plotted on a map. So the dark orange countries.
We can't hear you anymore.
Oh no. Oh.
Gabby.
Okay, thank you very much for telling me. Let me.
We can, yeah.
Bit better now. Oh,
Yes, I can hear her. Yeah.
yeah. Otherwise, please let me know. I'm sorry that there is this issue with that I have with Zoom at the moment. Yeah, please shout if I break off again. Okay, did you get where do I need to go back a little bit, or have you heard when I started talking about this slide? Okay.
We had you at the Federated EGA, so you can continue.
Okay,
Yeah,
great.
yeah, absolutely.
Good. Okay. So the dark orange are the countries that are currently full members of Federated EGA, with Canada over here. And those countries that currently have data available have this database sign here. And so at the moment, as I said, 5 nodes, we currently have 50 data sets. And yeah, and no longer restricted to Europe. So although we are still European by name, but we see us more as global by nature. And if you look at those 50 data sets that we currently have available, there is a large chunk of these data sets are cancer-related data sets. Then we have quite a few data sets in population genetics, some synthetic data sets, those looking at embryonic development, but also autoimmune disease and Parkinson. And I put the flags to show from which countries they come. And I explained the sort of data access process previously for the central EGA, but this is like a slightly modified version for Federated EGA, where so this is sort of the national node within a country. Then the data provider would usually reside within the same jurisdiction. They would submit data to their national node. All the sensitive information, genetic phenotypic data, would remain in this national node. And the data provider would also provide study level metadata, which is non-sensitive. And this would be transferred over to central EGA and would be presented in the catalog. So anybody who would wish to access the data would then be able to find the cohort information, the data set information in the catalog. They would then request the data access from the data access committee, which is set up by the data provider. And then once this is approved, the national node would then release the data. And so it's very similar to the original approach, just slightly modified for the Federated EGA. And so because we have so many countries participating in Federated EGA, we need a certain flexibility because there is variability in the national regulations and legislation around data protection. And so each node is then responsible for actually complying with all of this in their operations. So they set up their own governance and legal framework. They look after the data protection policies and make sure they have all the legal agreements in place to share the data. And additional variability comes in terms of what infrastructure already exists in the country, what is their funding and sustainability situation, and if a country would prefer more a central or federated approach to how their national node operates. And so usually the full nodes, all our nodes are currently full nodes, so they would accept data from external organizations, and which are then being shared through the federated network. but there is also an option for example, for consortia that have a data hub already set up, they could join the Federated EGA as a community node, which means they would not accept any data from other entities within their country, but they would just provide access to the data within their own data hub that relates to, for example, a consortium. And so this is a variation that is available. And we actually had discussions with Nikki Mulder and Mamana Bijavanga recently, who were thinking of potentially setting up an African community node within the Federated EGA network, which would be absolutely amazing if we could make this happen. And this was, I think, in the context of DSI Africa and AfroGen-D projects. And so this is a really exciting new development, and we are really looking forward if we could add a African node to the Federated EGA network. And yeah, this was my last slide, but I covered a lot of aspects. So these are all the people who were involved in the Recoded and Contagio project and the portals that I have talked about. And then obviously for EGA, there is also a whole team operating both in Barcelona, but also here in Kingston at EBI. And then obviously for the Federated EGA, we have to think about all our partners across the globe who work on Federated EGA nodes. And it's a great team effort to make this all happen. And yeah, and thank you all very much for your attention, and I'm happy to answer any questions or comments that you might have. And if you want to keep in touch with what's happening in EGA, we are available on various social networks, and also soon we'll have EGA newsletter that you could sign up for as well. So, okay. Thank you.
Thank you so much, Gabrielle, for that wonderful presentation on Federated EGA. We'll take questions now, or rather we'll just give 30 seconds for questions, which can be added to line 110 of the Framapad. And in case you have any just now, please feel free to unmute and just ask. We'll only take one so that we have enough time for Dr. Sumer Panji as well for this webinar.
Yeah, or you can always send me an email and I get back to you.
Okay.
I have a question, Laura, can I?
Yes, please. Sorry.
Yes. Thank you, Gabrielle, for the excellent presentation. I think it's good to see a lot of the work going into the EGA, and particularly around sharing of important data like the one you presented. My question was around highlighting this whole aspect of data sharing and the Federation in line with the EU Act of data. How do you triangulate that, given that this is very, I won't say delicate data, but it is, yeah, quite critical information. How did you manage to really maneuver some of the data governance issues at the EU level and also working across the continent? Yeah. Over.
Yes. So, so the way we address this, if you look at the how a data access request is processed. So we at EGA, we are purely data processors in the eye of the European regulation. So the European regulation defines different roles to people involved in sharing data. And so we are purely data processor, and we act on behalf of the data submitter, who remains the data controller. And they are setting up the data access committee. And so they remain in full control about who has access to the data. And we are just sort of keeping the data secure, and we only share the non-sensitive metadata about the data set. And initially, without the access request being approved. And then once it's approved, we can then release the data. And then with the federated EGA, so some countries have, in some countries, you are not allowed to submit data, for example, to central EGA. That would be sort of transferring the data across the border. And so with federated EGA, you have a national node, and all the sensitive information remains within the country. So, and therefore you don't need to share or send your data across a border to share it, but it will remain there. And then the national node, for example, could set up a trusted research environment where the data can actually remain in the trusted research environment, and the compute is provided to do the analysis. And that way, the sensitive data would never leave the country, and therefore it would comply with those stricter interpretations of the European regulations. Does that answer your question, Agnes?
Yes, it does. Thank you.
Okay, you're welcome.
Thank you, Agnes, and thank you, Dr. Gabrielle, for sharing with us. So now we'll, the rest of the questions, please add them to line 115 of the Frama pad. Please, Dr. Gabrielle, if you'd have time to just answer them on the pad, because we cannot take them now, as we will be welcoming Dr. Sumir for the second portion to highlight on African efforts. And I will introduce Dr. Sumir. He is the program manager of Elwazi Open Data Science Platform and the H3A Africa Bioinformatics Network. He has a strong background in bioinformatics and data science, holding a PhD in bioinformatics. Sumir has been instrumental in the planning, development, and implementation of significant research programs focused on building human and computational infrastructure, capacity, resources, and tools for bioinformatics, genomics, and data science within Africa. Welcome, Dr. Sumir.
Sorry, I was just looking for the mute button. Sorry about that. Thank you for that kind introduction. Can you see my slides?
Yeah, we can see them, but we are seeing them on speaker mode.
On speaker mode, okay. And is that better?
Yeah, that's better.
Okay. All right. Let me just quickly hide this display. Sorry, I've got two screens here. So yeah, thank you. As Laura said, my name is Sumir Panji, and thank you for Data Science Without Borders for giving me an opportunity to talk about some of the work we've done around FAIR and Federation. My talk will consist of two chapters. One is the chapter 1 of our FAIR verification journey, and chapter 2 is more around our Federation journey. Okay, not seeing these move. Okay, there we go. So currently, most of the studies on genomic data sets generated are very heavily skewed to European populations and genomes, which is kind of understandable as they're the early adopters of genomic technologies. There are some African data sets, but they form less than 0.16%, 5% of what's available, and they're usually African American in origin. Within Africa, as the Data Science Without Borders project knows, is that the living standards are improving within Africa. There's a prevalence of non-communicable diseases such as type 2 diabetes, stroke, and hypertension, and cardiovascular diseases, which are on the rise. But the lack of diversity does have ramifications. I mean, for now, a very good topic for people to research is polygenic risk scores that provide a risk estimate for complex diseases. and your risk of getting them. And globally, Africa has a disproportionate burden of communicable and infectious diseases, with HIV, TB, and malaria dominating the research on the continent. To ensure that Africa and African populations are not left behind in this global genomics revolution, the NIH and Wellcome Trust for the African Society for Human Genetics initiated the APHRC project or program, which funds specific African-based research projects dealing with African population, health, diseases, and genomics data. So just in a snapshot, APHRC has 48 projects with 15 collaborative multi-site centers for population genomics, 16 research projects, 7 ethics projects, 3 biorepositories, and 1 bioinformatics network, which was HGA BioNet. The APHRC consortium footprint is over 34 African countries and has 54,000 research participants with more than 500 consortium members. So HGBioNet is a consortium within APHRC that comprises of 28 nodes in 17 African countries with the mandate to develop bioinformatics expertise within Africa and provide bioinformatics support to the APHRC projects, and was led by Prof. Nick Mulder from the UCT Computational Biology Division. HGBioNet operates in a number of domains, mainly aimed at capacity development, that include providing bioinformatics education, training, provision of computing infrastructure, research, and computational tool development, as well as your data science, your data standards, harmonization, and workflows, and a lot more technical other aspects for that. We also function as a data coordinating center and archive for the APHRC genomics data generated for people to submit to us, and then we submit on their behalf to EGA. And on this right is just a quick overview of the types of data that's been collected by the APHRC projects. We have human genetic variation data and genomic sequence data. We have genotyping data, and we have microbiome data as well. So it's quite a wide variety of data sets. We tend to collect what we call metadata in this context is actually phenotypes. And a phenotype using the NIH definition is any clinical observation or measurement that's recorded and associated with an individual. This phenotype data can be considered as a significant part of the metadata being collected and are actually personal to that person. So these are some examples of what we are collecting as metadata within the APHRC project, such as your age, sex, your country of birth, current residence, native language, height, weight, current medications. And a combination of these phenotypes and genetic data makes it possible to identify different populations and individuals, especially if they have rare diseases and certain geographical regions. So that's why you need to have controlled access, similar to what EGA provides. So remit of the HGBioNet is to form as a data coordinating center for the APHRC projects. So we have a data archive where we have a whole process workflow over here where people notify us of the intent to submit data. They register on the dashboard and we assist them in preparing the data for submission. And we transfer the encrypted data to a landing area. We decrypt it and validate it. If we pass these validations, we map them to the EGA XML files, which are shown on your right, and then we transfer the encrypted data to EGA. And the nice thing about EGA, it does have a lot of fields that capture a lot of metadata information as well, such as your DAC accession, your policy accessions, your policies, your study accessions, and a lot of the other metadata that makes it quite nice and easy to find. In addition to the actual genomics data, we're actually also collecting, we have 3 biorepositories that are collecting biorepository samples from those actual data that have been generated. And those also need to sit in the biorepositories, and we need to provide access to them and registered access. So we've created an APHRC catalog, and we have 3 tiers of access. You have your public access, you have your registered access. You need to go through registered access if you want to request access for data. And we have access for the data biospecimen and access committee as well to approve or disapprove or reject applications. So I'm just going to move on to FAIR over here. And originally, the FAIR principles were published in March 2016, and sparked a major community effort by funders, researchers, just to understand the FAIR principles and how do we actually implement them. The experts shown over here is taken from a commission report by the European Commission for Research and Innovation, and it's called Turning FAIR into Reality, a final report and action plan from the European Commission expert group on FAIR data. Something to note is that the FAIR principles on their own are not enough. It's not just a checklist exercise, but you must also encompass the appropriate ecosystem to support researchers in making it fair, i.e. you must have an ethos to make stuff fair. So I was trying to make this interactive, so I'll put some questions over here, and I'm going to ask why it's very important. So any suggestions from the audience? Laura, are we allowed to be interactive, or do we have a bit of a time crunch?
Yeah, sure. We can be interactive and try and finish probably by 25 past. But
Okay, I'll
this
try
is
and
good.
go really fast. Okay, any suggestions or thoughts on the agents? Just type it in or write it in the fan pad. I'll give you 30 seconds and then I'll have a quick look at the chat. Okay, let's just go back to the presentation then. Okay, so there are many reasons. I mean, for researcher, it's important for me to be able to access data to continue my own research, which is great. But there's also an economic impact cost as well. So another report commissioned by the European Commission for General Research and Innovation from PricewaterhouseCoopers found that there's a cost of 10.2 billion euros every year for data that's not being made fair. Additionally, they estimate another 16 billion on top of that, which they cannot quantify. So you're looking at a cost of 26 billion euros per year for data not being fair. And if you think about this in the African context, where we are starting to really ramp up and generate data, it would also be it would also cost us as well if we don't start making data fair from the beginning.
Dr.
One
Samir,
interesting, yes?
I don't know if it's just for me, but I cannot see your slides anymore.
Okay. Okay, let me just do that. Let's see. Uh, okay, share again. Okay, can you see my slides now?
Yes, and we also had some answers on the chat about fair.
Okay, sorry, because I can't see the chat, so I should have thought about
Sorry.
this a bit better.
Okay.
One thing I do find interesting about this, sorry, so this is the economic cost I was giving about why fair is important. But what I really do find interesting by the authors of the study is they said they were heavily reliant on existing material, and if they themselves had fair data, they would have completed the study a lot more quicker. And I think this made them realize the importance of fair data. Okay, so this is another interactive question. I'm just going to skip it, but maybe just think about this as well. So we have different stakeholders. You have your research communities, which is us, and usually when they do create policies, it's left to the research community to implement them as well. But I think the nice thing about FAIR, it doesn't just apply to researchers. It also applies to data services, standards bodies, global coordination groups, your policymakers, your research funders, and also your institutions and publishers. So it's a whole ecosystem that's been changed there. The blue box at the bottom actually is a call from the RFA for HE-BioNet in 2017 for renewal. And here they asked us that we must make the HE Africa data conform to FAIR principles at that time. So these are just the FAIR guiding principles. You can see they're quite data-centric. You have your 4 indicators for findable, your accessibility, your interoperability, and your reusability. So we started a FAIR project within HE-BioNet, and this was led by Nikki Mulder and myself. And initially, we had a lot of discussion and policy development around FAIR at the start of HE-BioNet 2.0, which was 2017. We're lucky enough to contribute to some of the interim recommendations by providing input at the Science Data Conference 2018, when they had a session on critiquing the Turning FAIR into Reality report. Within HE-BioNet, we decided, okay, what do we need to make fair? So we did a bit of a scoping exercise to try and identify resources and outputs that require fairness to be made. And this is just a bit of the results that we got from there. We used questions from the Fair Shake rubric based on different resources to determine the following recommendations to make the resource fair. The results are pretty mixed depending on the resources, and the work done in harmonizing and mapping tautologies and submission of the HE Africa data would conform to FAIR standards, as well as for the tools and the workflows. When it came to assessing the HE Africa data, we used the Research Data Alliance Fair Data Maturity Model as a framework for assessing the HE Africa data sets. The framework consists of indicators which are mapped to the discrete components of the FAIR principles, which I just showed in previous slides. 7 are to do with findability indicators, 12 are accessibility and interoperability indicators, and 9 are reusability indicators. Within the framework, the importance of the individual indicators are classified into 3 categories: your essential, your important, and your useful. The useful is nice, it will help improve the fairness, but it is not mission critical, whereas the essential is mission critical. Without it, your data is not considered fair. The third component of the framework is how the indicators are scored to give a value, which can be improved on further iterations. This is just the way the sheet looks at like. The first column is the indicator mapped to the FAIR principle. The second column is a machine and human readable description of the indicator in the second and third column. The next columns are the priority of the indicator, whether it's essential, important, or useful. And there's also the score for that. There's another tab in the sheet that provides a more in-depth discussion or information about each of the indicators. So we took the data from the Asia Africa data catalog and we ran it through this assessment. And due to the information that required from the submitters and also due to the metadata standards and formatting that we have provided to EGA, and we helped them also complete any missing information during the submission process of the data sets to the archive, we're able to ensure a good data provenance and achieve almost 100%, well, achieve 100% fairness when evaluated against essential fair indicators. We have 76% coverage on non-essential indicators and an overall score of 87.8. To address the second row indicator, we have linked the data sets to initial publications and other publications that use the data set. So this is the indicator in row 2. And in row 3, we needed to refer to a standard reuse license. And this is very tricky because we have genomics data, and genomics data doesn't really have standard reuse licenses. So what we've done is use the data use ontology. So the data use ontology was developed by the genomics community to address the reuse conditions of genomics data sets that makes the conditions explicit. You have different categories such as your general research, your use within geographic region, or research ethics approval required. And this is quite useful for researchers that are coming in to apply for restricted access data sets because they can see what the conditions are and determine if their study falls within those conditions and then go ahead and apply for it, rather than going through the process of applying for the data and then be told, actually, no, your study doesn't fall into these conditions. And the DUO is also useful for data access committees that review applications and they can easily determine if the data request conforms to the reuse conditions. You can think of a Creative Commons license for genomics of population data. And the nice thing is because it is machine readable, there is a way to, the subsequent things is what we're looking at is how to automate the whole data access and request process using these tools. Okay, some other questions, but I think I'll just skip through this really quickly. FAIR, when people talk about FAIR, they tend to refer to it in the data context. And originally it was created for as a data context, but I think the report also indicates that fairness is not just the actual data set, it's also the provenance that generates the data set, your algorithms, your tools, your workflows, your analytical pipelines that lead to the creation of data and give it meaning. And this is quite important as I'll illustrate in the DSI Africa scenario. So one of the things that we have in Asia Bionet are workflows, which we created for the different Asia Africa projects. And this is to enable reproducible science and also automate science. I'm not going to go too much to what a workflow is, but basically it's just a description of a process, and it could be a pipeline, and it consists of a series of tasks. You have your starting task, and that task ends and completes, and then it initiates the start of another task. So this kind of like automates your pipelines and makes it a lot more easier to automate your analysis. If you've got like 10, 15 scripts, you can run it from start to end. And also makes it easier for other people to reproduce your analysis. Okay, so within the AGM, we had a bit of a FAIR and software workflows kind of session, and we decided to evaluate one of the new tools that was out there at that time, which is called How Fair Is. So the tool is a Python package that goes to your GitHub repo and does specific checks. It looks at your repository, looks at your license, looks at your registry, your citation, your checklist. And it uses like a 5-star rating system where you get 1 star if you're only compliant, and all the way up to 5 stars, which is green badge, if you're fully compliant. And this just shows you how to actually install it and how to run it. So I do ask you, if you do have some GitHub repos and you want to see how fair it is, this is a pretty easy software to use and do that. So we looked at what repos are actually active within Asia Bionet in terms of projects and decided to work on those repos. And this is just a screenshot showing us that for the Asia GWAS workflow and for the TADA, which is microbiome workflow, when we initially ran this package, we had 3 stars and 4 stars, basically. We worked on that and we got the tools registered in a tool registry service, which is BioTools. And doing that, and also doing other work as well. We managed to bring the overall fairness of the software workflows from 3 stars for the TADA to 5 stars, and the same for the HCA GWAS workflow as well. So the conclusion here is the limiting factor is not the data generation or increasingly multi-dimensional data, it's the data analysis and interpretation of the bottlenecks. And to apply tools and techniques such as your machine learning, you need well-labeled data. Curation of data and good data stewardship is essential towards adding value for data to enable findability and reusability. And I think for us during this journey, what we realized the fair landscape had changed from policy discussion to implementation with better metrics and tools now available for assessing fairness of different resources and infrastructures. And for us as a project, we got a better understanding and guidance on implementing fairness metrics for a range of outputs, which is now available. And with that knowledge, we're actually moving from making outputs fair to actually creating born fair outputs, which I think is quite important. And we're applying the skills and experience being gained to other African projects as well. So I'd just like to thank people from the HE Bionet grant and the FAIR project as well, and my colleague, Nikki Mulder. So I did have a bit of activities to try and make it interactive, but given the time, I think I'm just going to move on to chapter 2 really quickly. Unless, do we have any burning questions? Okay, so I'll just focus on the second part right now. So we are part of also APHRC Open Data Science platform, and that's part of the DSI Africa project. So DSI Africa project is harnessing data science for health and discovery and innovation in Africa. It consists of research hubs, your training grants, your ethical, legal, and social implication research grants, and the Open Data Science platform, which is APHRC. The DSI Africa Consortium has over 350 members, over 50 projects that range from climate change, demographic, data, imaging, genomics, mobility, and verbal autopsies, to name a few, and its footprint spans 22 African countries and has been funded for $88 million over a period of 5 years. APHRC itself consists of 7 African partners, 3 US and 1 UK-based partner, and its aim is to provide a flexible, scalable, open data science platform for the DSI Africa Consortium to find and access data, select tools and workflows, and run analysis on a choice of computing environments through easy-to-use workspaces. The platform leverages Terra and Gen3 implementations and of the data biosphere concept and incorporates Global Alliance for Genomic Health and tools and standards. So the main aim of APHRC is to develop an African open data science platform and associated resources. The solution needs to be feasible within an African context and aims to be flexible and scalable, enabling the implementation of data science for health. This is how we support the consortium through data, tools, computing, and training, but I'll focus mainly on the data working group and the computing aspects for this presentation. So APHRC aims to make use of the data biosphere concept, which should be based on 4 governing principles. It should be modular, which is composed of functional components with your well-specified interfaces. It should be community-driven by many groups to foster a diversity of ideas. It should be open source, developed under open source licenses that enable extensibility and reuse, with users are able to add custom or proprietary modules as needed. And it needs to be standard-based, consistent with standards developed by coalitions such as J4GH, just more for interoperability. This is an overview of the APHRC ODSP concept. So overall, a user should be able to search for DSI Africa dataset where possible, pull the dataset into a workspace, and be able to compute on it. If the data is not stored locally, then there'll be an element of either running the analysis by taking the code to the data, or if it's stored in the cloud, then you can access and compute the data within the cloud. So the data support working group's mandate is to enable greater data reuse across the DSI Africa by supporting the DSI Africa data hubs to share the data as part of the APHRC platform in the most interoperable form possible. The green box over here just shows the part that the data support working group has been working with. One of the sub-projects from the data support working group is to make the DSI Africa data fair. We also provide support in terms of data models, data harmonization, and have a catalog for searching the DSI Africa datasets. And this is still work in progress, though. We have the aims of working with the research projects for defining the conditions of data reuse, which is your geocodes from the consent forms, determine the fair maturity level, and work with them to improve the fairness of the datasets. We establish the fair goals for each of the DSI Africa data research hubs and projects and measure the baseline fair assessments at the start. By this time, there's been a lot more work done on how to implement FAIR principles and a lot more resources out there. So we decided to work with the FAIR Plus project, which produced a reusable verification framework for life science data. The framework includes tools, guidelines, and for making life science data fair. And the tools that we use mostly are the FAIR Dataset Maturity Model and the FAIR Cookbook. So the FAIR Plus dataset maturity model has 5 layers. You have your single-use data, which is 0, your identifiable data, your described data, your standardized data, your semantically typed data, and your managed data assets. So EGA will fall in category 5 of your managed data assets. The model is also described, comprised of three categories of requirements, which is your content-related, which is what is reported in the data set, and the metadata, your representation and format, which is the data object and metadata objects are represented or formatted, and your hosting environment capabilities. what capabilities does the hosting environment have that enables and supports the use of fair data, such as your APIs and all of that? To accomplish the verification across the device data sets and projects, we've decided to build a community of data stewards from the outset within DSI Africa. So each research hub has a data management and analysis core that's responsible for integrating various data sets for the project and has data management managers and stewards as part of it. So we've been hosting an annual data jamboree for the DSI Africa data stewards from 2023, which focus on the FAIR Plus framework and the verification and other aspects of data harmonization. We've continued this in 2024, where we focus more on fair data and the fair data maturity model as well. And we use recipes from the FAIR cookbook to help us improve the fairness of the data. It was a useful exercise in terms of increasing knowledge around fairness and building a community of data stewards. And also allows us to determine if there are any common FAIR cookbook recipes across the projects. This slide just gives a graph of what the verification goals are for each of the different projects and where they fit in. As you can see, the catalog Iliwazi has to accommodate all of them as well. This is just a quick snapshot of the catalog itself. DSI projects have registered data sets in the catalog, and it's still a work in progress. And we hope to have complete coverage of the data sets metadata within the catalog by mid next year. So the catalog aims to make the DSI Africa data more discoverable, and due to the metadata and data fields we're collecting and mapping to ontologies, make the data findable, accessible, and reusable. But still a nice illustration about how we build capacity around FAIR and how we're using it for other projects. As mentioned in Agnes's introductory video, there's a lot of diversity in data protection legislation, which is proliferating across African countries. And this makes it very tricky for cross-country collaboration, especially around demographic health and genomics data. This is a nice widget created by one of the DSI Africa projects that allows you to compare data protection laws against different countries. But if the data cannot leave a country's border, then we need to enable compute of the data on the local region, similar to the Federated EGA. So the infrastructure group comprises of research software engineers and scientists that aim to provide the computational infrastructure for the processing and computing of DSI Africa's datasets, and also to work to build a federated ecosystem. In order to build a federated ecosystem, interoperability and standards are crucial. And formed in 2013, the Global Alliance for Genomics and Health Consortium unites the international community to try and advance human health through genomic data. They build technical standards and policy frameworks and tools which can be used by the community itself. I'm just going to go through a couple of the standards that we're using very, very quickly. And please do look at the URLs to learn more about them. I'm happy to answer questions about them. One of the things that we need to figure out is, in order to analyze genomic data in the cloud or on-prem, a researcher must use an access tool to retrieve the file. But data repositories are crowded files, and as a result, the process for retrieving the dataset is complex and inefficient. To address this, J4GH has developed the Data Repository Service API, which provides a standard way to retrieve a dataset, regardless of the repository's underlying architecture. The next standard that we're looking at, which we're using, is the Data Connects standard. Data generators store the data in various formats, in structures, and locations, and this makes it very challenging for researchers to search, aggregate, and analyze data from multiple sources. It's also expensive for data providers to restructure the existing data sets to fit the requirements of highly structured, highly opinionated sharing systems as well. So as a result, a lot of data goes unshared and discovered. The Data Connect software developed by J4GH connects researchers to information and data sets regardless of how the data was originally stored or formatted. The next standard that we're looking at is the workflow execution service. So the reproducibility is kind of key to open science and was developed by the J4GH workstream. The workflow execution service describes a standard protocol for running the same genomic data analysis in different environments while still obtaining the same results. So basically, you can run the same compute in different locations, but you can aggregate the results and have this together to get an output. Another important component of this is actually the authorization and authentication process as well. The AI module enables secure data sharing by communicating the permissions needed to conduct research. So when looking at all these standards, it can be quite exhausting and quite daunting to actually be exposed to them and also trying to learn about them, as well as especially going through the documentations and trying to use them. We have the research software engineers within Iliwazi needed training and the availability of experts to deep dive into implementing the J4J standards. And we're doing this using the J4J starter kit. So this is just a screenshot of the technical hackathon we had in 2023, where we used a lot of these the standards which I described before to try and implement them. So out of that, we were able to run a pilot project to determine if you can use these standards to find data with a query and use that query to submit the analysis job to a web server located where the data is found. We had a starter kit installed on 4 sites within Africa as part of the pilot, which is in South Africa, 2 groups in Uganda, and 1 in Mali. And we used a region of 1,000 genomes and divided into 4 batches, so each one hosted on a different site and made accessible via the DERS servers. A user would query the Data Connect server, get a list of DERS IDs, and then select the DERS CRAM objects based on the IDs, and then submit these to the West endpoints, so each of the sites to compute on the data that's stored locally and get stats for each of those individual 4 files, and then aggregate the results into combined multi-QC. And this next slide just shows you the process flow of that. But in a nutshell, what we managed to do is actually use these standards to actually find data sitting in 4 different locations, compute on them independently, and aggregate the results, which is actually what federated compute is. We followed this up in 2025 with the hackathon again for GA4GH, and this was more aimed to move from the starter kit implementations towards production-ready implementations. The planning for this was quite inclusive, and at the start we had 15 members, but towards the end we had 40 people from around the world joining the planning meetings for each of the streams. And we had 4 streams. One was just the integration of the catalog, DNA stack, and data connect. Stream 2 was to identify gaps for integration and on-prem. Stream 3 was case studies of DSI Africa projects that have data in different countries and actually need to compute on them. And Stream 4 was a new stream for us. It was the implementation of GA4GH standards in trusted research environments. This is just a big outline of the program. So what we did was we packed the first day of the program with talks so we can get everybody on the same page. The next 4 days was actually just working on the hackathons. But what we wanted to do with this first day was just make sure that the streams are not duplicating work the other streams are doing. So that's why it was important to have the touch base at the beginning. And these are just the participants from that hackathon. There's quite a lot of work that was done, and there's quite a bit of enthusiasm. And we're actually lucky that we're working DNA stack that has a commercial implementation of the GA4GH standards and federated compute. And the plan is to integrate their software stack that runs on the cloud onto our on-prem compute facility to enable on-prem data discovery and compute, but also provide training to our research software engineers to enable knowledge transfer and skills development. This is the screenshot of what the interface from DNA stack looks like. And we managed to get some of the data from the catalog into there, but this is still a test case and it's still not fully functional, which is why we've not released it to the public. This is what we want to do in terms of putting it together is we want to make sure that we can have different sites around Africa that are able to host data, able to have an entry point where people can find data and run the compute and analysis on it and aggregate the results without the data moving. And in a global context, it's quite big because this data protection not only applies to us in Africa, but it applies globally as well. And we're finding it harder and harder to share data with different groups, especially in the global south, like in Singapore or Australia. And these kind of tools and standards and interoperability standards do enable us to work with other groups to ensure that data can be found and discovered and being used for research as it's intended to be, rather than siloed off. And with that, I'd just like to thank the Iliwazi members and teams and the PI, Prof. Nikki Mulder. Thank you. Oh, was my camera off the whole time?
Yes, it was.
Oh, no, I wish you told me. Oh, shame.
It's all right. Thank you for such a comprehensive talk on federated analysis, or rather sharing in Africa with the Loazi project. That was really good. I really enjoyed that talk. At this juncture, we're going to be taking questions, and that is on line 190 of the Framopad. But if you're able to just unmute and ask Dr. Sumer directly, that would be really, really great. Yeah, thank you, Precious, for posting on the Framopad. So, okay, I'll just go fast. I have a point of clarification, Dr. Sumer. There's a point where you said there's the GA4GH, probably I got that wrong, but is in 22 countries within Africa.
No, that would be the DSI Africa footprint as a consortium. We cover 22 countries in Africa. And some of those countries, research hubs have, similar to data science without borders, they have different sites in different countries. So for them to share data is quite difficult.
Okay. Do we have any questions?
Yes, please, if that's okay.
Yes, go ahead.
Sure. Thank you very much, Professor Panji, for a great talk. I have maybe 3 short questions. So the first one is on the availability of possibility of postgraduate students within Africa or Kenya to get access to some of this data from the previous H3 BioNet projects for reanalysis or even for testing a new hypothesis. Then the second question will be around data protection laws. For example, now we know about GDPR and some restrictions of data that can be sent out or into some of these countries. How that is taken into account in this, because I believe Kenya has some of the strictest data protection laws, it seems, on the continent, if implemented strictly how they stated in the letter. Then my final question will be to do with the commercial partners. We have all these biotech startups that are now generating this kind of data. Is there a way in which some of these will interface with the kind of data that are on these programs? And what if you could just shed a light on how that will play out? Thank you very much.
Great. Just happy to answer your questions, Frank. Just quick clarification. I'm not a professor. I'm a doctor. I'm not on an academic track. So the availability of access for data for reanalysis, yes, that is possible. So within, let me see if I can share my screen quickly and take you back a couple of slides. So we do have the H2 Africa data catalog, which people can request data from. So let me just quickly push that there. I think it's got the URL there. So they can just go to this URL and they can register and create an account, and they can browse the data sets and they can determine what data sets they need. So if they register, this is the view they'll see, but once they register, they should be able to see this view here. So yes, the data is available. You just have to request it through a data access committee, and that is available. Your second question around GDPR and data restrictions, how is that taken into account? So this is from your question, I heard you talk about data going in. So with this, what we're trying to do is make sure that data doesn't get out of the borders. People can compute on them and they can get results out, but we need to ensure that they get aggregate results out and not just individual level results out. So I think that's one of the things that we have to make sure that we do. You can go one step further than this and also then start looking at trusted research environments where before people can run the code on your data sets, it has to go through a sandbox where it's fully tested and the outputs are manually checked to make sure that there's no data leakage. And that's at its strictest level as a TRE. In terms of commercial partners, it's actually interesting because we are working with a coalition of pharma for the Genomics Centers of Excellence program, where we're partnering with them to generate more African genomics data and create data sets, which the commercial partners can have a copy of and the researchers can have a copy of as well. And I think that's a really good example of how you can have a commercial research and educational kind of partnership. Commercial partners are also welcome to apply for access to the data. If the data is under restrictions that it cannot be used for commercial purposes, then the DBAC will probably reject that application as well. I hope that answers your question, Frank.
Yeah, it does. Thank you. Thank you very much.
Thank you. Seeing as we already 5:30, any other questions, please put them on the Frama pad from line 196. I'm so thankful to everyone who's been able to join us for this webinar today. To Dr. Gabrielle and Dr. Sumer, thank you so much for taking your time to discuss with us on FAIR data and federated analysis and just shedding light on the global efforts and the African efforts that have gone into that. We're very thankful. For the participants, we will be sharing a post-webinar survey. Please, if you see it in your inbox, share with us your feedback so that we can be able to tailor this more and make the webinars more enriching for the community. we would have had a word from, okay.
To share material with us?
Yes?
Sorry for disturbance. Let me from Ethiopia. There is a breakup of internet. I can't able to follow all the presentations. So could you share material with us? So that
Yes, we will share the notes, the speaker slides as well, and the recording will follow. Thank you so
also
much.
we need the presenter's address. So present for them some questions.
Yes, you can put them on the Frama pad and they will be able to see them there. Unless they're comfortable with sharing their emails, then you can be able to contact them directly.
Okay, thank you.
Okay. Thank you so much, everyone. We will communicate when the next webinar will be, but for right now, thank you and bye.